Hawking Anna Shastraveta. He had done the protoplast isolation. Okay. Next, when you come again here, MS Media and manam chadutu untam anamata. So this MS Media was given by Murashid and Soog. So under their name, we call that medium as MS Media. So one thing you have to remember, students, what you have to remember is, manamu tissue culture చేసేటప్పుడు you say మనం MS Media use చేస్తున్నాం, Whites Media use చేస్తున్నాం, LS Media. So that is just the basic nutrient media. But అందులో మనము హార్మోన్స్ ఎంత కాన్సన్ట్రేషన్ వేస్తాము అన్నది ఇట్ డిపెండ్స్ ఆన్ యువర్ స్టాండర్డైజేషన్. సో Murashi and Shug or Blades Media or Whites Media or LS Media, any type of media, those are just the different types of media which we use, and in that we have to add the concentration of hormones what is required for that particular plant anamata. So itlane manaki we have angiosperms, we have gymnosperms, we have dicot plants, we have monocot plants, we have forest plants. So each one, before forest plants or the tree plants, tissue culture was very, very difficult. But nowadays we are doing lot of tissue culture plants in the forest plants also, that is tree species also they are doing. Okay. So next, clonal propagation, or in orchids, clonal propagation was done by Morel in 1964. Then haploids were developed by Guha in 1964. Haploid means pollen grain. Pollen grain as ante it's a male gamete. From the male gamete, they have developed haploid plants. Only male, and only from the pollen grain. There is no union. There is no fertilization between the pollen and the ovule. There was no fertilization. So from that, they have developed haploid plants. Then in 1970, Power, this scientist, he had done fusion of protoplast. సో మీరు అందరూ అనుకుంటున్నారు ఏంటి ప్రోటోప్లాస్ట్, ఎందుకు ఈ ఫ్యూజన్, ఎందుకు హ్యాప్లాయిడ్స్? బట్ ఈచ్ వన్ హిట్ హ్యాస్ ఇట్స్ ఓన్ ఇంపార్టెన్స్. హ్యాప్లాయిడ్స్ లో తీసుకుంటే యూ విల్ బి హ్యావింగ్ వన్ ఇంపార్టెన్స్, ఫ్యూజన్ ఆఫ్ ప్రోటోప్లాస్ట్ తీసుకుంటే యూ విల్ బి హ్యావింగ్ వన్ మోర్ ఇంపార్టెన్స్. దట్ సేమ్ క్లోనల్ ప్రాపగేషన్ తీసుకుంటే యూ విల్ బి హ్యావింగ్, ఈచ్ వన్ విల్ హ్యావ్ ఇట్స్ ఓన్ ప్రామినెన్స్, ఈచ్ వన్ విల్ హ్యావ్ ఇట్స్ ఓన్ అప్లికేషన్స్. ఫర్ ఎగ్జాంపుల్ నేను చెప్తాను, haploids are there. So when it is haploid plants, what happens is you don't have the two things and ovular and pollen grain fertilization has not happened. Suppose if fertilization happens, what happens? You will be having embryo development. From the embryo, what you will get? You will be getting the seeds anamata. So haploids lopala emoutundi ante what they do is colchicine treatment ichesi they will do the what you call they will double the chromosome and you will be getting the plant anamata. So the antropala kuda, if you get fruits, what will happen is you will be getting the seedless variety of fruits manaki ostund anamata. So you have the seedless plants, or you if it is haploids, you will be having different types of what you call a leaf developments, different characteristics atlanti you will be finding. Suppose protoplast fusion, and you take two different plants, one will be having disease resistance, another will be having high yield anamata. So you take both the protoplast, you fuse both of them, so it will become a single protoplast. So a single protoplast, you will grow the plants. So what are characteristics you will get? You will get both the characteristics you will get. You will be getting the disease resistance and you will be getting the high yield, both the things you will be getting there. So each one, it has its own what you call applications it has towards the mankind or for the development of the science. Okay. See, then in vitro cultures and ankunna. What are the in vitro cultures? Ante what are the explants we can use ani that we'll be discussing. So first is a culture from the intact plants. Ante you can take seeds or seedlings cultures you can take. Then embryo. So embryo always it should be immature embryo anamata. So if you take the immature embryo, what happens is you will be having little bit of what you call meristematic activity manakandulo kanipistu untundannamata. So you can take the immature embryo, you can take. Then you can do organ culture. In the organ culture, you have the shoot tip culture, You have the root culture, you have the leaf culture, and the anther culture. So many cultures are there. Then again, you have the calyx culture. I told you from the explant can either directly get the root system and the shoot system, or in between you can have a stage called as a calcing stage. So callus culture also you can do. Then cell suspensions, usually for the secondary metabolite productions and all, you'll be having this cell suspension cultures another matter. After that, you have the protoplast culture. So many different types of cultures are there. All these cultures are done under in vitro conditions. In vitro conditions means, keep in mind, it is under the laboratory conditions. Laboratory conditions and it has to be having the nutrient media undali, it should have the hormone system, and over all the above, it should be under the sterile conditions. Okay. Next, you have few applications of the tissue culture. One is the micro